Hello, it's so wonderful to see everybody today. As Amanda said, I'm Bianca Russell. I'm a clinical geneticist at UCLA, and I've been involved with the ASXL community since 2013. And so it's really amazing to see where things have gone and all the things that we're doing now. So really excited to talk to you a little bit about one of the big goals that's come out over the last couple of years for the community. And that's really about this concept of what do we do when there is a treatment, a drug that our community can access? How do we make sure we're ready for that? And so that's been a big focus of our clinical research team. And you'll hear from both Natasha and I during this talk. And she's been a huge part of really bringing that online and taking us to the next level. So I'm going to start by just sort of introducing some of the big concepts here and going through some of the definitions about different types of clinical research that all feed into the goal of developing a medication for treatment. So this is a kind of schematic that I made that took me a while because I kept making arrows in various directions and they went all different ways. And then I finally said, enough, we'll just make it as simple as possible. But the big picture here at the top is if we want a medication for treatment, how do we get there? And we have to get there through a clinical trial. And there's a whole arm of drug development involving our basic scientists. We are not talking about that part of it today. But what we want to focus here in purple is on what we do to get the clinical community ready for this. And this requires developing measures, biomarkers, natural history studies, biobanks, and a real concerted effort to have clinical endpoints that we can monitor in a clinical trial to truly show if a medication is safe and effective for all of your families. So what is clinical research? And most of these definitions you'll see have come from the NIH. And so clinical research in general is the study of people or data or samples of tissue from people where we're trying to understand a health condition. It helps us find new and better ways to diagnose and treat medical conditions. And it's often done by doing clinical trials, which is a way for us to test new treatments for a condition. Natural history studies, which we'll talk about in a little bit, are an important way for us to collect health information to better understand these conditions and how they change over time. Patient-centered research outcomes is one of the kind of buzzwords that we've been spinning around the clinical research space and has actually come to the forefront as being one of the really important foundational pieces of the work that we do in clinical research. And the idea here is that we're not just doing clinical research to do it. We're doing it with the idea of what matters to the patients, what matters to the family, how do we make sure that it's not a siloed effort by the medical team to make decisions that make sense to them and answer their research questions, but also include the research questions of the families and making sure that together the focus is on the patients and the families' beliefs, opinions, and needs in combination with the medical community. So one of the big tools that we use in clinical research are natural history studies. And the idea of this is, as I said, sort of understanding a lifetime, right? So what happens over somebody's life who has a specific condition? And this is a really important concept for our community. And having taken care of many families clinically, it's usually the first questions people have. What do I expect? How does the future look for me? What's going to happen as my child gets older? And the idea of a natural study is that it's observational. So we're observing the natural course of a condition, and we're trying to understand over time what happens in that condition. You can study other variables, things like what treatments people are using and what medications are being used, but it's not interventional. So we're not actually, the goal is not to treat the condition, it's to watch it and to understand over time what happens individually and through the community. And another really powerful tool that we use in clinical research is biobanks. And the idea here is that we can collect human tissues. It can be all sorts of different tissues. anything you can imagine, basically, collected for research purposes. And the idea is that we store these over time. So sometimes we collect samples and have a very specific goal for them, and there's a research study or a question that we want to answer by looking at those samples. But many times the concept is we have an opportunity to collect these samples, we're going to collect them, and we're going to store them in a repository. And over time, as we develop more questions, we'll have this resource and this tool that we can continue to tap into. So it can really serve as an incredible resource for the basic science community, who is also working on these questions and is a critical part of what we're doing to develop drugs and to answer those questions about why the ASXL genes are causing these conditions. So those samples sometimes have immediate uses, but oftentimes are really stored long-term. And we only in the future come up with questions to ask of those samples. Another really important component of getting something, of being able to assess really effectiveness of a potential treatment, is biomarkers. And these are a couple definitions I found that I thought were pretty helpful. They both kind of cover different components of it. But the basic idea is there's something objective, so something not subjective, not variable based on lots of different things, but a really objective measure that can be an indicator of sort of a normal biologic process, an abnormal biologic process, or even a pharmacologic response to something that we're doing. So if we're giving a medication, so if we note that the brainwave activity is abnormal in an individual, we give them a medication, we see normalization of that brainwave activity, that could be a really important biomarker to say, okay, what we're doing is making a difference here. And this can be really any substance, structure, or process that you can measure within the body to help understand the condition. And we'll talk a little bit more about this one later. And then the grand sort of finale, when you get all these pieces coming together and when you've got your basic scientists who come up with treatments, and you've got the drug development team that's got a drug that they're producing, and you've got all of the clinical people who've said, okay, we know about this condition, we know the history, we know what families need and what makes a difference to families. We have outcome measures, we have biomarkers, we're ready for it. And so clinical trials is where we actually take a medication to the community and work to make sure that it's safe and effective. And this is a treatment to humans with an intervention. We typically have an arm or a part of the group that gets a placebo, so not the medication, and another arm that gets the treatment, and we compare safety and efficacy of that intervention. Okay, so that was our sort of intro about all the things. I'm going to pass it off to Natasha here to talk a few more.
Okay, so I'm going to talk a little bit about some of the information that the Federal Drug Administration, the Food and Drug Administration, has kind of given us in terms of how we kind of prepare for clinical trials. Okay, so we're held to very, very high standards, right, to get drugs approved. It's a huge process. And over the last decade, there's a real recognition by the FDA that we really need to include the patient voice in that process. And that makes complete sense. You know, these treatments are targeted for these individuals, and we need to include their voice, their families' voices in that process. And so there's a new guidance series that the FDA has published to help guide those who are developing drugs, those who are developing these natural history studies that are going to be used eventually for drug approvals. And these guidances are very, very helpful. So the first two guidances really focus on helping researchers and industry really understand how to conduct the research, to understand what's important to patients to treat and therefore measure in a clinical trial, right? And then there are also some guidances about how best to measure those things, which can be complicated at times. But again, the emphasis is throughout this process, including the patient voice through that process. So really guidance 1 and 2 focus on developing these what we call concepts of interest. So things that are really important to treat and to measure in a clinical trial. So these concepts of interest are really aspects of an individual's experience or clinical, biological, physical, or functional state that a clinical trial outcome measure is intended to capture. And again, the emphasis on this really has to be important to patients. So these guidances tell us we need to do a lot of qualitative work. We need to talk to families, do interviews, do focus groups. It's one of the major reasons that we're doing focus groups at this conference today, to start to refine these concepts of interest that are most important to you all here. Guidance 3 is really focused on, okay, well then how do we measure those things? Measurement can be really complicated. There's many ways you can measure things. You can have patients, share how they're feeling directly. But for some of our kids in the ASXL community, they may not have that ability to share effectively in a verbal format, right? And so we have to find other ways to try to get information from them, right? We also rely a lot on observer reports, so caregiver reported outcomes and measures, right? So I'm sure many of you here have filled out questionnaires as part of your child's clinical evaluations or in school. So we definitely want to look at those as well. Clinician reported outcomes. So this is what, like, if you go see a provider, kind of what they may rate about your child or write about your child on certain scales, right? And then performance outcomes, so direct assessments. So your children may have experienced some of these neurodevelopmental assessments, cognitive assessments. So there's a whole bunch of ways that we can really get and measure these concepts, get at and measure these concepts of interest. But in our community of rare neurogenetic conditions, particularly ASXL related disorders, there's often some barriers to using kind of the standardized measures that we have in our toolkit. That's because these children are really complex, right? So they may have severe motor impairments. They may be nonverbal, right? They may have vision difficulties. They may have hearing difficulties that really impact the validity or how useful some of these standardized assessments are. Some of you may have had developmental assessments from providers or your school educational teams, right? And I have heard from many families that, those measures just don't capture what my child's full potential is, right? So there are certainly limitations, and we need to start solving those problems. And that's what I'm really passionate about. That's what my research team is really passionate about, is figuring out how to modify or develop measures in a way that's really going to capture, what your kids can do, right? And to capture those concepts of interest that are important to you all in an effective manner. All right, so guidance 4 really focuses on endpoints. And so this term endpoints is essentially what is measured ultimately in a clinical trial. So the measures kind of measure, different aspects of the individual's functioning, how they think and feel, how they're able to function throughout the day. But endpoints is actually the number or the statistical kind of data point that we need to capture to then compare, the treatment arm versus the non-treatment arm. And I think an emphasis here in the guidance is that these endpoints really need to be meaningful to families. And then, how to capture, guidance 4 also focuses on, you may have a certain degree of change that's statistically meaningful. Is that change meaningful to families though, right? There's a difference between whether something's statistically significant when we do all the fancy statistics, right? And whether that translates into something that's meaningful for families. Additionally, we may see change that's not, when we do all of our fancy math, right? It's not statistically significant, but a family may notice, like, no, there was this change and it was completely meaningful for my family, right? One example that I've heard from some of the rare disease communities that I've worked with is head control, right? So if a child's unable to hold their head up, right? But then let's say they make some progress there in therapies or with treatment, they're able to hold their head up, right? Well, now that opens their world, right? Now they're able to make eye contact more and see their peers, right? It opens up a whole new level, a whole new world for them, right? And a whole new skill set that they can then work towards. So but that skill may not be measured by some of the standardized assessments that we have, or when we apply the statistics needed, that might not be captured. And so we really need to do a better job of making sure that those small but meaningful changes are captured. Okay, and this is kind of just a big schematic kind of showing this whole process across those guidances that I just talked about. We're really still kind of in the red and green in the ASXL related disorders. We're still learning about patient and caregiver perspectives. That's why we're doing these focus groups, right? We're still identifying these concepts of interest that are important most to you all. And so we're going to talk now a little bit about some of the research that we're doing that's really informed by what we know about what we need to get ready for clinical trials through natural history studies and these guidances that FDA has provided for us. And so I'm going to start off talking about some of our PCORI work or our focus group work that we're doing at the conference. So this all started back in 2022. The foundation put forth this effort to create a research roadmap in ASXL-related disorders. And you may remember they sent out a survey that asked several questions about, what's important to you in terms of research within the ASXL-related disorders. I pulled out one question from that survey. They asked, which symptom or challenge is most important to you that we have to try to alleviate or improve? You all were amazing and really rose to the occasion. In literally 2 weeks, completed 205 of these surveys. So we learned so much from you in that short period of time. And thanks to the foundation and Amanda in particular for really pushing that survey out. And we are just really happy to have this data because it really set the stage for many of the things that we're doing this year at the conference, and is really going to help move forward the needle towards clinical trial readiness. Okay, so we got many responses from 28 countries. And this is really a summary of the data. This was a poster presentation presented at a conference in 2022, and it really describes kind of the top areas of priority for families in terms of treatment. And so it differed a little bit based on ASXL 1, 2, and 3, but kind of the overall theme is that really neurodevelopment, so communication, cognition were really important. Also behavioral difficulties, and then also gastrointestinal symptoms. So this really, we really wanted to learn more about what families meant though, when they said, oh yeah, communication is a concern or behavior is a concern, GI is a concern. We want to learn more about what exactly within those domains is of most importance. And so the foundation, so Amanda and I teamed up, and we are co-PIs of a grant we submitted to the Patient-Centered Outcomes Research Institute, which is a funding agency who really focuses on patient-centered outcomes research. We applied to this grant, and we were successful in obtaining it. And so this grant really focuses on engaging families in the research process to really understand what is most important to families. So we were awarded a grant for $100,000 this year. So it's happening now. And this is really, there's two aims. One is to really build education about why it's so important for families to be partners in research and to participate in research. And so we really have been doing that throughout this whole year. I kind of mentioned this already, but the first aim here is really to educate, you guys kind of here in this context about the importance of your voice in this research. And then also we have been sort of meeting throughout the year with a group of representatives from each of the disorders. We have an expert in each disorder. We've been meeting monthly throughout the year to really talk about what we mean when we say patient-centered research. And this team has really been integral in developing kind of the way in which we're conducting the focus groups that you all are participating in at this conference. And the second aim, of course, is the focus groups themselves, right? So we really want to focus on understanding the priorities of patients and families within those top 3 domains. So neurodevelopment, GI, and severe behavior. And so we're going to be doing that today. Yep. So I talked about this already. One of the main kind of tenets of our group, we call it a topic group team that's met throughout the year to really work together and collaborate in developing this focus group project. Everyone's voice is equal, and everyone's voice is equally valuable. Everyone has a seat at the table, whether you're a caregiver, whether you're an expert, whether you're a scientist. We all are working together, and we are all collaborative in this effort. So the focus groups you participate in today was a collaborative effort among patients and families and experts and scientists. So we've met throughout the year. We're conducting focus groups now in Baltimore. After the focus groups are over, we're going to start to look at our data that we collect. We're going to summarize kind of the main themes, and we're going to start to prioritize some research questions that come from that. And then we'll be sending out a survey to you all to really rank, in your perspective, what are the most important things for us to study and to target in clinical trials. So do you want to switch? Yeah. So now Bianca's going to talk a little bit about some of the natural history studies going on in ASXL related disorder.
Okay, great. So building on that, we talked a little bit about natural history studies being a really important component of clinical research and clinical trial readiness. And so we're really fortunate to have several different clinical research teams who are interested in our community. And we heard a little bit yesterday from Dr. Emily Woods about the ASXL3 natural history study in the UK. And through that study, she's collecting detailed medical histories on individuals with Bainbridge-Roper syndrome. Simon Searchlight has also been a part of our community for quite some time now, and they are also specifically collecting information on ASXL3, and they have standardized questionnaires that they collect across their network. Their network is specifically interested in genes related to autism, and they have biological samples as well that they collect. So they have sort of a biobanking component as well. And then, since I'm the one who's up here talking, you'll hear more about what our work is, but we also have traditionally had a registry at UCLA that we're really excited to be rebranding and sort of relaunching as the ASXL Related Disorders Natural History Study. And so it's been a long process for us. The registry started around 2016-17 when I was a resident at Cincinnati Children's, and it's been really fun to see that project grow. And I'm really proud of what we've done to be able to feel like we have longitudinal data, long-term data, and data points that we're going to continue to get for the ASXL community. And so I've sort of launched this as our natural history study. So a little bit about what our study is. It's a multi-site study. So UCLA is what we call the coordinating center. The IRB is sort of our ethics board that in academics helps make sure that everything we're doing is ethical and that we're protecting all of the families that are involved in research. And so UCLA holds that sort of ethics approval for this study. But what makes it so spectacular is all of our collaborators. So these are all of our sites here and what we call PIs or principal investigators at each of those sites that are closely associated with us. These are some of our enrollment numbers here by ASXL condition. And thanks to this conference, we've been growing quite a bit. So we're really excited to have so many new families joining our group here. One of the foundational elements of the natural history study at UCLA is a collaboration with families. And we've gotten some fabulous feedback from all of you that you guys wanted to be more involved in what we're doing. And we're thrilled to have that happen. Over the last year and a half or so, we've developed a steering committee, which meets quarterly and twice yearly. We open it up to all of you. And we hope if you haven't joined us that you will consider joining us virtually for some of those meetings in the future. The members of the steering committee include family representatives, external researchers sort of outside of our network, statistician at UCLA, and the UCLA clinical research director who really oversees a lot of the research programs at UCLA and helps us with a lot of the administrative processes that are involved in regulating research. And the purpose of the steering committee is really to help us stay on track. So make sure that we're still in line with what matters to all of you and to the community, make sure that the goals we set are actually happening, and help with communication to make sure we're communicating to all of you the meaningful things we're doing. And then the committee itself also reviews requests for data access. So one of the elements that's really important to us is that this community remains open and collaborative, and that any data that's being collected is accessible to the research community. If they're asking appropriate questions and have the right tools, we really want to make sure that they have access to all this information that you've been so willing to provide to us. And so one of the things we've done to kind of build this natural history study is to increase our collaborations with other specialists. And so Natasha's been a major collaborator, and she's going to talk a little bit about the ALP study, which we're collecting here, as well as Dr. Judah Wilson from UCLA and her champion study, which many of you have been involved with as well. And then the biobank, which is another kind of component of the natural history study. So I think I'm going to pass it back to you, Natasha, right? You're doing this part? Okay, Natasha's going to talk about some assessments within the natural history study.
Okay, so I talked a little bit about how it's important to utilize assessments and measures in capturing the natural history of ASXL related disorders. And so we're working, of course, with our collaborators like Dr. Russell, Dr. Judah Wilson, and others in terms of doing projects where we're collecting this information. And so, you know, we're really trying to do this in a standardized way because, as I mentioned earlier, we're under tight restrictions and guidance, right, from FDA. And so we want to make sure we're doing this the right way from the get-go. And so I also wanted to mention too, we're focused a lot on clinical trials today in this talk, but a lot of what we're doing is also informing clinical care now, right? So we're learning a lot in the moment and along the way. The process, you know, to drug approval is a long one, but along the way, we're learning all this valuable information from you guys, both through the qualitative work and some of this more quantitative work, these standardized assessments that we can translate, you know, even in a quicker way to, you know, learn and disseminate to you all kind of sooner than drug approval, right? So we're really excited about that too. All right, so first I'm going to talk a little bit about the ALP study, which is a study that I'm leading. So it's called the ASXL Behavioral Phenotyping Study. And this is, again, kind of a sub-study of Dr. Russell's natural history study. And so we're really focused on understanding the behavioral phenotype of ASXL-related disorders. So when I say behavioral phenotype, what I mean is really the characteristic cognitive, functional, behavioral, and psychiatric pattern that's typical in a disorder. And this is really important for the natural history study. We can start to understand, you know, how the different gene variants relate to, you know, how a child phenotype. So what I mean by that is kind of what they look like, right? Like how they're functioning, you know, their thinking skills, their communication skills, those sorts of things. Any medical pieces as well. What aspects of neurodevelopment and behavior are part of the disorder and may be important to target for treatment. And this can also help us understand what measures may be best to use in clinical trials. So the ALP study is a really big effort, and this is, you know, 3 parts. And so it contains assessments on cognition and communication. We're including a neurological exam and motor exam. We're looking at autism symptoms because we know that kids with ASXL related disorders are at higher risk for autism. And then we also are doing a functional vision assessment, which really gets at cortical vision impairment or difficulties in processing vision. We're starting to learn that children with neurodevelopmental conditions may be at higher risk for this. So as part of data collection here at the conference, we're doing some of those assessments. Some of you may have done them already in your time here. We're also taking more information from caregivers through doing interviews on adaptive functioning remotely. So this is how a child is functioning through the day. And then also about problem behavior. We learned right from that survey in 2022 that problem behavior, disruptive behavior is really a priority. And so we want to learn more about what that looks like in this community. And so my colleague, Dr. Julia O'Connor, is going to be doing some remote interviews regarding that. And then there's a questionnaire packet as well. So some questionnaires are going to be collected on paper here at the conference, which you may have already gotten when you checked in. And then some will be done remotely in many different areas that are of priority as well, like sleep and quality of life, of course, that's extremely important. And also some information to learn from you about caregiver family functioning and unmet needs. So we're really excited about this ALP study. And again, all the data is really going to go into the registry, right? So it's we're going to be able to share among collaborators. And so we try our best to not have you all repeat things, you know, around the same time. We want to reduce burden as much as possible. We know already how much of a commitment and we're so thankful for your participation. We're learning so much already. I have my research team who's working with you guys, and they are all just I think I've recruited them to this space. They are so, first of all, they love all your children, and they're just learning so much, and they're really intrigued, and they're making comments and coming up with new questions and things to think about for this community, which is just so amazing. I'm like, guys, write it all down, and we're going to meet later and talk all about it. So it's been really, really fun. Also, I'll touch on Dr. Rajuda Wilson is one of our colleagues at UCLA, who's also working. Her particular interest is in motor development and how that relates to autism and neurodevelopmental disorders. And so she's doing a lot with gait mapping. So some of your children may have participated in a gait mat where you kind of walk across a gait mat and it looks at kind of the pattern and catches really subtle differences in motor skill. So we're really excited to merge that data together with ALP study and of course what Dr. Russell collects through natural history study. So lots going on.
Okay, we're almost there. Final handoff. Okay. So as we talked about, biobanks are a really important part of clinical research and a really, really powerful tool integrating what we're doing clinically with the basic science side of things. And what we've actually found as well is that the power of the biobank is matching up the clinical data. So being able to have biologic samples from individuals and also having neurodevelopmental information, medical histories, other biomarkers, all those things together create an incredibly powerful resource for the research community. And so one of the things we've done at UCLA is the samples go through a close collaboration I have with Dr. Arboleta and Dr. Wexberg. Dr. Arboleta is at UCLA and Dr. Wexberg is at Sick Kids in Toronto. And in a combined effort, we created a public website called the REACH Biobank. REACH stands for Rare Epigenetic and Chromatin Disorders. So it includes the ASXL conditions along with several of the other chromatin-related genes that are studied in both the Arboleta and Wexberg labs. And this is a place where you can actually go and explore. It's intended to be designed to help the research community understand what samples we have and what data we have and make that accessible to the larger community. So we're hoping that this will help recruit researchers to come and study our conditions and learn and be able to access this information. So the website itself serves as a sample inventory. We list the information that we collect in the natural history there so that researchers know what types of questions we've asked of the community. And then there's instructions about how researchers can submit a proposal to access this data. So I encourage you to take a look at that website. And we're so thankful. I think you were able to collect over 30 samples yesterday from your families. So thank you to all of you who are willing to donate samples. And we're really excited to see this grow. I also do want to point out that Simon Searchlight also has a biobank. I sort of mentioned that before, just for ASXL3, but that again is another really powerful resource. And many researchers studying autism use the Simon Searchlight database. So another great way to get your samples out there and continue to allow researchers to sort of ask these questions. So one of the last concepts we have here is biomarkers. And so this is something that's pretty new to our community, but I think really exciting and quite powerful. Many of you have been involved in our EEG study for boring open syndrome for ASXL1, where we were doing in-home EEGs on your children. And one of the outputs from that study was to look at what's called EEG spectra analysis. So this is a little different than a clinical EEG where they're really looking at the brainwave patterns and looking for seizure activity. This is more of a research tool where we're looking very specifically for some markers that have been associated with certain genetic conditions and that can be used over time to say, okay, this is a marker of what individuals' brains look like who have ASXL1 related disorders. And then the idea that in the future, if there's a treatment, this is what the changes that we can see on EEG. So this image here is just an example. The blue line are sort of the typical individuals, neurotypical individuals, and the red lines are children with boring open syndrome. And what you can see there is in between that 6 to 12 hertz range, those two vertical lines there, there's a clear peak in the blue line. And we call that the alpha peak. And that's what we expect for children of this age. But when you look at the red line in that same area, you don't see that peak. And this seems to be a marker that is specific to individuals with Boring-Opitz syndrome. It's also seen in other genetic conditions, but it can be a really important biomarker. For example, if we see an individual does not have this alpha peak, you treat them with a medication and you see they start to develop that alpha peak, that may be a really early indicator that what you're doing is making a difference. So this has been a really exciting study and something we're hoping to expand to the rest of the ASXL community in the next couple of years here. Another biomarker that's been developed and continues to be worked on is to look at the methylation patterning. So we can take blood samples from individuals and in collaboration with Dr. Wexberg lab at Sick Kids, we've been able to identify that individuals with ASXL1 have a very different patterning of what genes are turned on and off in their body compared to their neurotypical controls. And so what you see here on the graph, the purple circles are individuals with Boring-Opitz syndrome, and you see they all sort of cluster together, and then the controls in gray that are clustered separately. The other map here with the blue and the yellow, what this is really showing is the initial part there where you see the purple at the top, those are the individuals with Boring-Opitz syndrome. And you can see that they have almost an opposite pattern, right? They have yellow on the top and blue on the bottom versus the controls that have an opposite pattern. And so what that's really showing is that there's a difference in the blood in how the genes are controlled. And that's very specific to Boring-Opitz syndrome. So that again is another marker that if you were to provide a medication and you saw that then this pattern reverted to the control pattern, could be a really powerful marker that what you're doing is making a difference. And again, this is something we're working on for ASXL2 and 3. Okay, so then the final grand finale, right? Clinical trials. We're not there yet. But we're really excited by the framework that we're building, and we're feeling really optimistic that we are creating and working really hard to get our community ready for clinical trials. We've talked today about patient-centered outcomes, natural history studies, biobanks, and biomarkers as really important components of that process, and really excited about the progress that we've made. I want to sort of finish up here by telling a story about one of our sister conditions, which I think has personally actually really inspired me as a clinical geneticist and been really wonderful to see treatments being developed for neurodevelopmental conditions. And I think the story is something that resonates with our community and the things that we're working towards. So Rett syndrome is another chromatinopathy caused by genetic changes in MECP2. It's also a neurodevelopmental condition. And this is a condition that we've known about in genetics for a very long time. So they've had a very long-standing natural history study, and the course of the condition has been well described over many decades. They also have an incredibly strong family foundation that's been driving research and supporting basic science and clinical research for many, many years, and that's the International Rett Syndrome Foundation. The family community is incredibly engaged, much like all of you, and they really worked hard to develop clinical trial endpoints with family-driven, meaningful change goals, and developed a Rett syndrome specific survey that was used in their recent clinical trial. And so in March 2023, there was a new medication called trofinetide that was approved by the FDA specifically to treat individuals with Rett syndrome. And when I went on the website to sort of grab the logo and get the information, they had released a letter to the community marking the 1-year anniversary of the drug's approval. And they reported that over 1,300 people are currently being treated with this medication, and they reported some of the data showing, of course, some variable success, but that it has certainly had an impact on the community. And I think that story is really powerful and something that I hope our community will get to. It may not always be that we can find a cure that's going to reverse everything, but like Natasha mentioned, finding the meaningful change, what that is, and hitting that milestone is really significant. And I know it's had a dramatic impact in the Rett community and honestly, the whole genetics community, to be able to see that there's something that's sort of impacting and changing neurodevelopmental conditions like this. So just as an inspiration moving forward, we are not the first to tread this path, but it's really exciting that I think we are certainly making our own way here and coming up with things that really make sense for our community. So we want to just finish by saying thank you to all of you. Thank you for being our partners in this. Every person in our community matters, makes a difference. We can't do it without any of you. And I think it's really important that all of you get a chance to be involved in any capacity that you want. So please don't hesitate to talk to any of us. There are many of us sort of in the research community who are here amongst you today. Please pull us aside, share your thoughts, your insights. Help us make sure that we are getting our community ready for clinical trials. So I want to thank you all for your attention, and I look forward to connecting with people with the rest of the meeting.
You guys want to come up here and we have a couple of questions. We have 3 minutes for questions, then we're gonna get ready for our next session. So this is your chance. Pull out your phones, scan those QR codes. We do have a couple that have been coming in through the while we were getting going here. So gonna have you, yep, green light, and then lean in really close so we can the people at home can hear you. Okay, so the first question is, do the natural history enrollment numbers include the recently enrolled conference participants?
I don't know, Julia back there, you gave me those numbers pretty recently. I think they did.
They're almost accurate. There were a couple last minute additions.
A couple last minute additions, but pretty accurate. One of the things I think that is important to note when enrolling in the research studies is we do have to go through a pretty rigorous process of reviewing genetic testing, the consent process, making sure that everyone who is joining the research study really qualifies for those studies. So we know that sometimes that process is slow and it does take some time, but it is important in the long term that we're really being thoughtful and making sure that those that are being involved in studies, truly qualify for that. So it does take longer than just sort of starting a Facebook survey or something like that, but it has an enduring value.
Thank you. So the next question that has lots of votes, and this may be better answered in tomorrow's basic research session, but maybe we can introduce it here. And that is, how is research looking with CRISPR technology? If you can offer a few words on that question.
CRISPR is an amazing tool being used in the laboratory to study genetic mutations and to cause genetic mutations in cells to be used as a research tool. It's not being used at this point in a meaningful way in humans. So I know our research community is using that as one of their research tools, but it's not something we're looking at utilizing in humans.
Thank you. The next question is, what is the medication that can cause the alpha peak to be reached for Boring-Opitz syndrome? I think that's in relation to the slide that you showed with the EEG study. Does that question make sense?
Oh, I think what they're getting at maybe is that if we were to develop a medication that we could see somebody who doesn't have an alpha peak then develop an alpha peak, and that would be a marker that treatment was working. So we don't currently have any medications that change the alpha peak in Boring-Opitz syndrome.
Thank you. I love this question. What would you say to a caregiver who is scared by the quote unquote research study moniker? How would you encourage them to participate in research?
Let's talk.
I think that's really healthy, right? So I think it's really important. It's your role as a caregiver and as a family is to protect your loved one. And it's actually really important that you are a little nervous, right? So that's actually a really important sentiment, and means that you're being thoughtful and thinking about what's happening and how to make sure you're protecting your loved one. So we really encourage that. And I would say, if you're nervous about it, talk to us about it. Ask us what your questions are, what are your concerns, and how can we help you feel better about it? And if you get to the end of it and you still don't feel good about it, there's no obligation to do any of it. Or you feel good about it initially and then you change your mind, that's okay too. So it's really important that anyone involved in research feels welcome and feels comfortable with what we're doing. And if you're not comfortable, it's really important that you express that.
Yeah.
So we really do encourage you to communicate with us about what you're feeling.
I definitely agree with that. And I would say that after talking, there may be parts of it that you're comfortable with, and there's parts of it that you're not comfortable with, and that's okay. You can participate in part of it. That's totally reasonable. So yeah, definitely come talk to us. I think we're always willing to have those kinds of conversations.
Thank you. So we'll end it there. We're going to move on to our next session. There are a couple of questions that I will go back and try to answer through text. So you can keep an eye on what you have up on Slido, including one about how far we are from clinical trials. And our Chief Scientific Officer, David Batoon, will be here tomorrow. He's going to talk about that a little bit in a talk also with Dr. Bayless from University of Michigan. So we'll be able to answer that a little bit more in depth in tomorrow morning session. So thank you both, Dr. Ludwig, Dr. Russell. Appreciate it. Thank you.